I'm Kyle Gears, and this is Thursday Brain Bytes. So, past couple weeks, we've been talking about recording electrophysiology in the brain of model organisms. We've touched on how electrical potentials are created by neurons, how different frequency bands of neural activity have relevance within the brain, and then we've talked about some of the cells, neurons, oligodendrocytes, astrocytes, microglia, that are in the neural environment and how they interact together in the nervous system to form the brain. So today we're going to talk about how neurons respond to having electrodes inserted next to them. So we need to keep in mind when we're performing electrophysiology, it's an invasive measurement and we're actually perturbing the neural system around this. So I'm going to address three specific papers, one from the Kozai lab and then two from Aaron Purcell's lab at Michigan State that are investigating the effects of implanting silicon microelectrodes in the neurons and how those microelectrodes affect the neural environment immediately following insertion, as well as in the intervening weeks. So before insertion, using two-photon microscopy, we can look at neural activity using optical calcium indicators. And so this is a measure where calcium flows into the cell and your cell will glow green. So prior to insertion of electrodes, you'll have kind of the ability to visualize the vasculature. However, immediately following electrode insertion, there is a propagating wave of neural activity known as cortical spreading depression. And so this is calcium flowing into the cells and indicative of cortical damage. About a minute after the insertion of the electrodes, there's still an area around your microelectrode array that have increased intracellular calcium levels. And so as a major finding of this study, the Kozai lab was able to demonstrate that insertion of these microelectrodes can cause tissue deformation and tissue damage that leads to rises in intracellular calcium, and that these intracellular changes in calcium affect neural activity in the immediate period following electrode insertion, as well as for about a week following electrode implant. Building upon this work, the Purcell lab has looked at the morphology of neurons near and far from electrodes. And so when you have a naive pyramidal neuron, right, it has this really nice complex structure. However, when electrodes are examined near the electrode, you actually see a simplification of dendritic spines and the dendritic arbor. So this means while cells are there, they become less complex. This decreases their ability to receive and interpret signals and can alter the overall excitability of the signal. And this is actually only true within the first couple hundred microns relative to the cell. However, we have to keep in mind that when recording electrophysiology, those signals are biased within this range of 100 to 50 microns from the electrode shanks. And so we're recording electrical signals from these neurons which are affected by the presence of the recording shank. Further work from the Purcell lab then did RNA-seq experiments to examine how changes in RNA expression correlated with these changes in neuron morphology. And what they were able to show is that following electrode insertion, there's destabilization of the cytoskeleton and an increase in microtubule motor proteins. So these are the proteins that are thought to be transporting proteins in mitochondria into the axons and dendrites of neurons. However, during this upregulation of cytoskeletal and microtubule proteins, there is a decrease in the overall synaptic stability proteins. So we're able to show that these calcium-mediated changes in protein expression are reflected in the overall cellular morphology. And we can then relate that back to the work from Eles et al. In 2019 about the overall propagating calcium waves that are seen in following electrode insertion. So I'm Dr. Kyle Gears, and that's your Thursday Brain Bite.